Morning everyone, welcome back. I would like to introduce Mendelian genetics. After looking into genes, chromosomes, let us now start by discussing Mendelian genetics. So Mendel is the father of genetics, so it's known as Mendelian genetics, but by his name is known as Gregor Mendel. He did his work in the year of 1822 to 1884, and he was a monk, meaning he was more like a bishop in a church, but he was also a scientist or a researcher. He did a lot to make us understand much of hereditary science, and we appreciate. And it makes us to understand much how the traits are inherited from the parents to their offsprings. So Gregor Mendel is the scientist who is responsible for the laws which govern hereditary science, or which govern inheritance of traits. And if we talk about the laws, as we proceed in the other slides or the other sessions, we'll be able to see each of those laws, the three laws which govern the inheritance of traits. And it still talks about inheritance of traits, bearing in mind that at the time we were introducing the genes, we said the traits are the phenotypes, and behind the phenotype there is a gene which is responsible for such phenotypes. So in this case, Gregor Mendel in 1822 to 1883 did a lot on genetics, and he's the one responsible for the laws which govern inheritance of trait. So he is known as Gregor Johann Mendel. He was an Austrian monk. So the first point, he studied inheritance of traits using pea plant. Pea plant is in the class of the kundes. I don't know whether it's the kundes, but the cowpeas. So he did his research or his study of inheritance of traits using the pea plants, and that is where he developed the laws of inheritance. So his work was not recognized much until in 20th century. That is when Mendelian's work came to light. So between 1856 to 1863, he cultivated a lot of the pea plants, 28,000. He cultivated and tested them for us to get much to understand of the three laws that govern the inheritance of those traits. So he found out that the plant's offsprings were retained. There were offsprings which could retain traits from the parents. So as long as the offsprings are the generations from the parents, so he saw some traits in the offsprings which were seen in the parents. And at that point is where the laws or the pea plants will help us to understand the laws of Mendel on how these traits were inherited by the offsprings from their parents. So Mendel, at the time he was doing his research or his work on inheritance using the pea plant, he stated that the traits or the physical traits which were inherited were particles. For him, he recognized them as particles, but he didn't know that actually the traits which he refers as particles were actually the chromosomes and the DNA, because we say it has to start from the DNA. The DNA resides in the genes. The genes resides in the chromosome. And at the point of the crossover, that is the point where the traits are inherited by the offsprings from the parents. So recombination is involved. So we describe much structure, organization, function, as well as variations, the origin of variations in population. So the particles which Mendel stated to be inherited by the offspring from the parents were actually chromosomes and the DNA. And that is why we had to look at the history of the concept of the genes using different scientists, how they understood it. But at last, whatever we saw was crowned by the central dogma theory. So Mendel had to add to it so that we could understand the three laws, how all these three laws were applying for us to understand the hereditary science. So the particles which Mendel tried to refer as the physical traits which Mendel tried to refer to as particles were actually chromosomes and the DNA. So for us to understand the genetics or the hereditary science, we'll be able to use a punnet square. A punnet square is going to help us to solve all the problems in genetics. Be it you had asked about the monohybrid cross, the dihybrid cross, you go to population genetics, you asked much about calculating the phenotypic, the allele frequencies, the genotypic frequencies. For the multiple alleles or the biallelic situation, we'll be able to sort it out. So the punnet square is used to help in solving the genetics problems. And remember, the punnet square, in the punnet square, we've already said for the gametic, the sperm will donate half, the other one will be donated by the ovum or the egg. So you have to present them in a punnet square or on a punnet square so that it can help us to solve the problems. So that is how a punnet square works, how to make a punnet square. So first, what you do is you draw the columns, and in the column, the first row, based on how the statement says, so we are going to say what it means to have the capital Y, what it means to have the small y, what the capital Y and the small y represent. Of course, they are alleles, the alternate forms of a gene. So you'll have the parent 1 here, the parent 2 here. The parent 1 means it can be the male, which has the sperm. Parent 2, it can be the female, which have the ovum. So you can arrange them the way you want, but up there you have parents, here you have the parental generations. Then, just like you do the matrix in mathematics, you'll see if this parent contribute the allele, the dominant allele, and this parent also contribute another dominant allele, then in the first box you will have the homozygous dominant. So we are talking about the genotypes at this point. So you have the alleles. The alleles were the alternate forms of the gene. So if you talk about a trait, for example, let's say the seed shape, it's either wrinkled or smooth. But then you talk about the capital S, because the smooth will be dominant over the wrinkled. Whenever you are trying to work on your beans, you want to put beans, you want to cook the beans, so you'll always remove the wrinkled. Even when you want to plant, you use them as seeds to plant. You'll not take the wrinkled ones, of course you'll take the smooth ones. So it means the smooth will represent the dominant allele S, but the trait is the seed texture. That is a trait, a gene controlling the trait seed texture. You are looking at the phenotype, you are saying this is wrinkled, this is smooth. But what does smooth and wrinkled represent? Texture. So there is a gene which controls the seed texture. But then in that gene, there is alternate forms of that gene. So it means the dominant allele will carry the capital S. The recessive allele will carry the small s. Don't put capital S and maybe capital W for wrinkled. No. It is not dihybrid. It is monohybrid. We are looking at one trait. We are using the Punnett square to help us solve it. So for example, in this case, they are talking about the seed color. We are using the Punnett square. But remember, we talked about the homologous chromosome. Each of the parent has to contribute. The male with the sperm will bring, if it's human, sorry, the female will bring. So in that case, we are going to have the capital Y for the yellow color, but still the gene controlling the color. You've seen the color as the phenotype, the gene product at the level of protein synthesis. You can see the color, the phenotype which is expressed, but then behind that phenotype there is a genotype. So the gene which explains why the color Y is dominant, the capital Y, it will give the dominant allele. The capital Y represents the yellow color. It means the small y, the recessive allele, it can't be expressed when the dominant is already expressing. It will only be expressed when you have both of the y's, small y's. We are talking about the recessives, homozygous recessive. So in this case, the recessive allele will be small y. But they are the alternate forms of those genes, the gene which controls the seed color. So the Punnett square, based on how the statement is, you'll have the heterozygotes. Both parents are heterozygotes. You'll have the capital Y for the dominant allele for yellow, small y recessive allele for green, because green is recessive. It's not expressed only when it is a homozygous state. Both of the alleles are small y, small y. So we talk about homozygous recessive. So in this case, the genotypes for the parental generations are heterozygous. Capital Y, small y, for one of the parent, it could be from the mother side, and then the father side, parent 2, capital Y, small y. So we talk about these two parents being heterozygous. Their genotype is heterozygous. So in the first box, Punnett square help us to solve all the genetic problems. In the first box, you'll have capital Y, because one of the capital Y's is contributed by the parent 1. Another capital Y will be contributed by the parent 2. So you will have a genotype of capital Y, known as homozygous dominant. Then in this grid, the second row, you will have capital Y, small y. It means for this, the small y was contributed by the parent 1, and then the capital Y was contributed by the parent 2. So you have an heterozygotes here. Then here, you just follow the way you could do matrix in Form 4 or Form 3. You have capital Y contributed by parent 1, parent 2 will give us small y. So you have heterozygous. Then lastly, we'll have small y contributed by parent 1, small y contributed by parent 2. So you talk about homozygous recessive, whatever you are seeing here. Meaning, we are talking about the genotypic ratio, the genotypes. Genotypes will always be homozygous dominant, heterozygous, and homozygous recessive. Full stop. So here you have the F1s, whatever you see in these 4 spaces in here. These are parental generations. These are parental generations. Parent 1, parent 2. Those are parental generations where with their genotypes, each of them is heterozygous. But then at the point now where we have the F1s, the generations which come from the parents, you can see it has to pass something down the line. The first offspring, by chance, came out as yellow. 2 capital Y's. The law of dominance. We are going to discuss that when the laws come. So this offspring, by chance, we have the homozygous dominant. We have heterozygous here, which means still the law of dominance. The capital Y suppresses the expression of the small y. The dominant allele will suppress the expression of the recessive allele. So in this case, the phenotype will show as yellow. Still the law of dominance. The same with this one. And then lastly, we will have homozygous recessive. Each of the parent is going to contribute the recessive allele. So in this case, we talk about the phenotype. It will appear green. The homozygous recessive, they are only expressed when both alleles are recessive alleles. But in the case here, it can't show the green color because already the allele for dominance, the yellow, was dominant over green. So it will suppress the green color such that the phenotype in this case will appear yellow. So that's why you can see the phenotypic ratio is 3:1. 3 yellow, 1 green. And when you're talking about the phenotypic ratios, you have to mention the phenotype. Don't just put it 3:1. 3:1 what? 3 yellow, that is the phenotype. Because there was a genotype which underlined that. You saw 2 of the heterozygotes, 1 of the homozygous dominant. Definitely the heterozygotes were going to express the color yellow, which was dominant. So in this case, we talk about the genotypic ratio. Genotypes are always homozygous dominant, heterozygotes, and the homozygous recessive. So you will have 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. So in this case, we have genotypic ratio of 1 capital Y to 2 capital Y small y to 1 small y. In this case, the phenotype will be 3 yellow, 1 green. Sorry, I went ahead of introducing you to the genotypes, but I was explaining how the punnet square is going to help us understand the genetics questions or problems and help us to solve it or to solve them. So a bit of the terminologies which we are going to mention every now and then. We are going to talk about traits, any characteristics that can be passed from the parent to offsprings. Remember, that was the definition of genetics, and remember, recombination and mutations are involved in this. And still remember the central dogma theory. At the point you're seeing a trait, at the point you're seeing a phenotype, there is a genotype which underlies it. And then a bit of the hereditary as terminology, passing of traits from parent to offspring. And that's how simple it is. It will involve the 3 laws of Mendel, but then the basic idea here is we are passing traits from the parent to the offsprings, and then genetics is the general study of hereditary. We are going to have several crosses to understand much of the laws. The monohybrid cross, it involves 1 trait, e.g. it can be flower color, seed shape, flower height, maybe the color of the skin if it's human being. Yes, so it involves only 1 trait. Dihybrid cross involves 2 traits. So it could be flower color combined with height. It can be seed shape combined with the position of, maybe the position of the leaf, for example. It could be axial or terminal, the position of the leaf. So it is involving 2 traits, that is the hybrid. Then we have alleles. We talked about the alternate forms of the gene. In this case, we're talking about the 2 forms of a gene. We either have the dominant as well as the recessive. So Alleles are the two forms of a gene. A gene, I said, it can control the flower color, the flower height, that it can control one trait. A gene controls that one trait. But in this gene, the trait which we are looking at, there is a contrasting characteristic. One is tall, the other one is short. So there has to be a allele representing the tall, the other one has to be representing the short. That is if you're looking at a trait based on the plant height. Then the dominant one is always stronger of the two genes. It is the one which is always expressed, and it is represented by the capital R. And then the recessive one is the one which shows less, and it's represented by a lowercase, which is small r, or the smaller letter. Genotypes, we talk about the homozygous dominant, the one which has the dominant allele, capital R, for example, capital R and small r, the heterozygotes, small r, the homozygous recessive. Then the phenotype, that is the physical expression or the physical feature resulting from the genotype. Just as I was defining the genes, I still repeat that it is a phenotype. A phenotype will always result from a genotype. There has to be a genotype for you to get the phenotype. So the phenotype will always describe the physical expression. So you can talk about the red color of a flower, the white color of a flower, the blue color of a flower, or the tallness, the shortness. That is the phenotype we are talking much about. So we have an example of genotype, generating the genotypes based on their alleles. So in this case, the red color is dominant over the yellow color. We are talking about the flower color as a trait, monohybrid cross. We are trying to generate a monohybrid cross. So at that point, we are talking about the flower color, controlled by that one gene. But that gene will have alternate forms, will have the alleles, which will represent the dominant allele for the red, the recessive allele for the yellow. So all the genes will occur in pairs, will have two alleles. And the possible combination of the genotypes, I said the genotypes will be homozygous dominant, heterozygous, and homozygous recessive, full stop. So the phenotypes, when you see the capital red, that is homozygous dominant, they will show a red color as the phenotype. Then capital R and small r will still show the red color because the dominant allele representing the red color will suppress the recessive allele for the yellow color. So whatever will be expressed is the red color. So those flowers will appear red as much as the genotypes, they are heterozygous. Then we have the small r, the homozygous recessive. They will clearly show the yellow color because both alleles are representing the yellow color, which was recessive. Homozygous genotypes, when they appear all capital or all small, we talk about homozygous genotype. But we will talk as well as they are also known as pure breeds, because whenever we have the capital and the small one, the dominant and the recessive combined in heterozygous state, we talk about the hybrid. But in this case, they are pure breeds. We have pure, capital, all dominant alleles, it is homozygous dominant. All recessive alleles represented, then we talk about homozygous recessive. So the gene combination involving two dominant or two recessive genes is known as the homozygous dominant. Sometimes it is referred to as a pure breed. And then when we talk about heterozygous genotype, it's a combination of one dominant allele as well as one recessive allele. And we have the capital R, small r. In the case of the example, we talked about the color of the flower, the red and the yellow. Remember, the red was dominant over the yellow. The yellow was the recessive. So in that case, it's known as hybrid. So this is the case of a phenotype. You'll be able to see the color of a flower. It appears pink, it appears white, but that phenotype, that physical expression, whatever you can see and conclude that it's based on this color, it's based on this shape, it's based on this texture, that is the phenotype. Behind that phenotype, there's a genotype, which explains the existence of such a phenotype. So Mendel used pea plant, and he grew it. It was grown in a small area. Remember, there were 24,000. The advantages of the pea plant, or the Pisum sativum, for the people who are good in taxonomy, it could produce lots of offsprings. If it produced lots of offsprings, it was easy to trace how the characteristics or the traits were inherited per generations. So after the parental generations, we had the F1s, the F2s, until the generation which Mendel wished to clearly see how the traits were passed. So it could produce lots of those offsprings, the generation that is. And it also produced pure plants when they did self-pollination. So that is what Mendel did. It was self-pollinated, and it will give us several generations. And this could be done by artificially crossing the plant. So Mendel produced pure strains. Remember, we talk about the pure strains, the homozygous, dominant, homozygous, recessive, allowing the plants to self-pollinate. So after having the parental generations which were pure, then for those generations there was self-pollination. So from the F1 you do a cross again of the F1s, and then you have the F2s until the generations which Mendel will always generate, so that we can understand much of the genetic or hereditary science. So it could self-pollinate for several generations, and that is now when we got to know about the traits, how they were inherited from the parents. So this just shows how the self-pollination and how Mendel did his work until he would get the traits for all the generations which he had. So these are the traits Mendel did. He worked on seed shape, seed color, pod shape, pod color, seed coat color, flower position, plant height, flower color. 1, 2, 3, 4, 5, 6, 7, 8. 8 traits. Mendel did 8 traits. So for the seed shape, the gene controlling the seed shape had the 2 alleles, round and wrinkled. The dominant one was round, and it will get the capital. The wrinkled one was recessive. It will get the lowercase one, the small r. Seed color, yellow, was dominant over green. The gene controlling the seed color had 2 alternate forms of the genes, that is the alleles. Allele responsible for dominance, the yellow color was dominant over green. So the yellow color was capital Y, and the green color was small y. Pod shape, smooth, dominant over wrinkled. So capital S for smooth and small s for wrinkled. And then pod color, green, was dominant over yellow. Seed color, coat color, was gray, dominant over white. Flower position, axial, was dominant over terminal. So the terminal will get the small a, as the dominant one. Axial will get the capital A. Then plant height, the tall one, was dominant over the short. The tall will get the capital T, and then the short will get the small t. And lastly, the flower color, the purple, was dominant over white, and so the purple had the capital P and the white had the small p. So those are the traits which Mendel studied. So this is what the outcome was. So it showed the wrinkled, smooth, dominant allele, recessive allele, the seed shape as the trait, and then you get the alleles, and then the physical expression, whatever you could see, the wrinkled, smooth, yellow, green, inflated, wrinkled, green, yellow. So you'll be able to see those are the traits which Mendel worked on. We are talking about following the inheritance of these traits from the parent. So the first generation which we start off with are the parental generations. Sometimes they're referred to as P1, but they are parental generations. The parental generations in a breeding experiment. So they're the P1. Then the ones which are generated at the parental generations are the first filial generations, the F1. So the first generation offspring in a breeding experiment, they are known as the F1s, or the first filial generation. So from breeding individuals from the P1 generation, you will get the first filial generation. Then the second filial generation is from the F1s. So from the F1s, you'll get the F2 generations. Now you'll use the genotypes of the F1s to get the F2s. That's how it goes. Then the F3s, you'll use the genotypes from the F2s so that you can get the F3s. So it shows how the generations are generated. You have the P generation, the parental generations. In this case, it clearly shows the plant height as a trait. So you have the tall one, the capital T, pure. We talk about the pure plants. Down here you can see the genotypes representing the pure parental generations. So you have the tall, the homozygous dominant, capital T, capital T. You cross it with the short one, small t, small t. So you have cross of two pure plants, and it will result into hybrids. We are going to see that because the Punnet square is going to help us solve that. So parent 1 is capital T, capital T. Parent 2 is small t, small t. So you will have a generation of all heterozygous, all hybrids. Then from the F1s, you'll generate